we should have done the intro, so if you did do it, go ahead and turn to page 8.6 and I'll stamp it really quick. But while I'm stamping it, again, I did tell you I was right on the back, it said do page 8.6, do Wednesday, Thursday. But while I'm doing this, I want you guys to look at this picture, okay? I want you to say, what the heck are we looking at? Can you get this in? Did you finish it? Yeah. Okay. Again, I want you guys to see that this is literally the whole thing of cellular respiration. Where's my cup? Matthew just turned it in on Sammy and Matthew was right. Just turn it in. Take a picture tonight or tomorrow. Okay, we just sent it. So again, this is everything. We're not going through every single chemical reaction. We're not looking at every single piece. I'm showing you this because I want you to have a little bit of appreciation for me cutting it down into the condensed version that we're about to do. Okay, so just understand, I'm saying this as nicely as possible. It could be worse. Okay, it could be worse. So I'm just cutting it down to the bare necessities, because if you take further future biology classes or whatever, then you'll learn more. If not, then that's fine, because this is all you'll ever actually know. And for most of you, this will be the only time you ever see cellular respiration in your entire life. And that's okay. Okay. Again, that's why I'm hitting the highlights and not going in depth. Okay. So let's go ahead and start with this part. So aerobic cellular respiration. So if you would go ahead and read it for me, please. Actually, I'll read the first sentence. Cellular respiration is the vital process that allows organisms, both heterotrophic and autotrophic, to convert glucose into a usable form of energy called adenosine triphosphate, or ATP. So how can we compare and contrast heterotrophs and autotrophs using the word glucose? How can we compare and contrast heterotrophs and autotrophs using the word glucose? What do autotrophs do with glucose?
They store it.
Yes, but before they store it, because technically we do too. They make their own glucose in a process called photosynthesis or chemosynthesis. So one more time, compare and contrast heterotrophs and autotrophs using the word glucose. Autotrophs make their own glucose, while heterotrophs consume glucose. But regardless, what do they do with that glucose? Turn it into ATP. So that's question number 2. Off to the right. It says compare and contrast heterotrophs and autotrophs. So when we compare them, we say, or when we contrast them, autotrophs do what again? Autotrophs make their glucose, while heterotrophs consume their glucose, but both use glucose to make there. Perfect. Okay, so starting off with ATP is composed, if you would, go ahead and read it for me, please, Sam.
ATP is composed of a nitrogen base, adenine, ribose, and 3 phosphates. In the presence of oxygen, this process is called aerobic respiration with another inorganic molecule that is not oxygen. The process is called anaerobic respiration.
Right there. So, looking at this first part, it says that ATP is composed of a what? Which is called? It's composed of a sugar called ribose, because anytime it ends in ose, it's a sugar. Just like if it ends in ace, it's an enzyme. And how many phosphates? 3. So, looking at question number 3, let's label it. So, so what is this part right here? Nitrogenous base. What is this part right here? The sugar called ribose. And what is this part? The phosphate. But looking back at the notes, it says that after the parts of the ATP molecule, it said that if oxygen is present, what do we call that type of cellular respiration? If oxygen is present, what do we call that type of cellular respiration? Such as an aerobic respiration. And if oxygen is not present, what do we call that? Anaerobic respiration. I go back to this previous slide. Again, aerobic respiration means oxygen is present, anaerobic means oxygen is not present, but what is the? What does it say? An inorganic molecule. What does inorganic mean again? But what does that mean though? Inorganic means that it's not organic, but organic means that it's composed of this and this. Hydrogen and carbon. So inorganic means there is no carbon, there is no hydrogen. Okay? Such as metals. Okay. Go ahead and read that last part where it says if the final. Read it for me, please. Abigail.
If the final electron acceptor is an organic compound, this process is called fermentation.
Has anyone ever heard of fermentation before? What is fermentation?
It's like when you preserve it.
Like when you preserve it, and things like sauerkraut, say what? Wine, beer, kimchi, soy sauce. There's tons and tons of products, food products, all across the world that where people have learned to ferment to produce a specific type of food. But what does that mean? That means they're literally doing anaerobic respiration, where the final electron acceptor is not, or is what?
Inorganic.
Is an organic molecule. So instead of oxygen, like us, like everyone else, it's some type of organic molecule, which ends up producing some type of product. For example, when you ferment alcohol in wine or beer in wine, the final electron acceptor is a molecule called acetaldehyde. And the end product, instead of water, is alcohol. So that's how you end up producing wine, and end up producing beer. Okay. Does that make sense? Okay. Go ahead and continue reading on for me now, please.
Aerobic respiration involves 4 stages to break down compounds and store energy in electron carriers. NADH and FADH2 to be used in a osmotic process. So really quickly before we move on to looking at the rest of this, NADH and FADH2 are what we call electron buses. Isabel, go ahead and sit next to Matthew on the other side. So that way you don't break your neck. Sorry. We'll move everything there. So again, electron buses are NADH and FADH2. Anytime you break something, just like we talked about last week in chapter 7, whenever there's a chemical reaction where we break bonds, energy is released, right? Remember, that's an exergonic reaction. But that energy is stored in electron buses. One more time. Anytime we break something in this chemical process, we release energy, but it is stored in a chemical bond with electron buses. They store it. And they're gonna deliver it somewhere, just like how buses deliver people to different stops, stop A, B, C, or whatever, okay? So, what is the point of aerobic respiration again? It says aerobic respiration involves how many stages?
4.
To break down who?
Compounds.
But what compound? What's the first one that we start off with?
Glucose.
Glucose. It always starts with glucose. Again, all of our conversations last, the first and second test, kept talking about glucose, okay? And we're still talking about glucose. Go ahead and look at questions 1 and 4, because we already did 2 and 3. Actually, we'll do one together. Aerobic respiration requires what compound? O2. Number 4, match the following compounds with their process. Well, we just kind of did it, but aerobic respiration requires who? O2. But what about anaerobic respiration? What does it say in the notes? That's true, but, anaerobic respiration, it says, uses inorganic molecules. NO3. Remember, inorganic means it's not organic, which means no carbon, no hydrogen, which that's nitrate, so it doesn't contain anything. And then fermentation, in the notes it says what? Who's the final electron acceptor? Some type of organic molecule. Well, which one of those is the only organic molecule? CH3CHO, whatever that is, okay? So it should be C what?
A.
And then B, perfect. Let's look at glycolysis. If you would, go ahead and read it for me, please. Again, we're starting off with the whole process, okay? Go ahead with glycolysis, Savannah, please.
Glycolysis occurs within the cytoplasm, and its purpose is to break down one glucose molecule, 6-carbon sugar, into 2 molecules of pyruvate, each 3 carbon.
So what is the point of glycolysis?
Breakdown.
It breaks down glucose, which is how many carbons? 6, into 2 molecules with how many carbons? 3. So we're basically just cutting it in half. That's all we're trying to do. But it's a little bit more complicated because we got to do some stuff to it before we can actually cut it up. Okay. So how do we start off? We'll start off by go ahead and Savannah, read the next part, please.
Glycolysis starts by adding a phosphate on carbon 1 and another phosphate on carbon 6 to produce fructose 1,6-bisphosphate.
Bisphosphate, good.
This process requires 2 ATP molecules.
So what's going on? So essentially we end up getting, or starting with who? What molecule do we start off with? Just for clarification, I know this is hard. I know it's a lot of names. I know it's a lot of molecules. I know that. And it's boring, okay? I know that. This is something that we have to learn, but I am condensing it, and the only way we're gonna somewhat get to it is by rinsing and repeating. That's the only way that we're gonna do it, okay? On next Tuesday, we have a Kahoot where it's just question after question that asks, where does this happen? How much is produced? Because if you remember the exam, a lot of those questions were, most of them were kind of like critical thinking questions, like the one about the muscle, or what does it say? It said something about smooth ER, which one of these organs has more smooth ER? Like that was a hard question. For chapter 6 or 8, there's no question like that. It's just, it is what it is. There are 4 ATP that are made. There are 2 NADH that are made. There are 2 pyruvates that are made. There's no critical thinking involved. It's just, you gotta memorize it, and then if you do it, great. If you don't, you don't. Does that make sense? So, just bear with it, okay? So anyway, going back to this, what is the first thing that we do? We start off with glucose, which is how many carbons?
6.
6. 1, 2, 3, 4, 5, 6. But we end up with 2 molecules of pyruvate, which are 3 carbons. We start with that, we want this. So how do we do that? The first thing that we do is we add phosphate at the front and phosphate at the back. So phosphate here, phosphate here. But to do that, what does it say? It says add another phosphate and carbon 6 to produce fructose 1,6-bisphosphate. This process requires what though?
2 ATP molecules.
So essentially, I thought we were supposed to make ATP, not use it. But this process is a little weird because it does require a little bit of energy to kind of kick it off. It's the same thing as like whenever you're trying to start a lawnmower with like the handle, you know, you get down, you gotta pull it really hard. It's kind of the same way. You gotta kind of have to put energy in to let the lawnmower to go. Okay, so to do this, it requires what? 2 ATP. We gotta use 2 ATP. So we start with glucose. We add a phosphate at the front, phosphate at the what? But since this is this glucose still?
It's fructose.
It's not because we added phosphate to it. Now it's called fructose 1,6-bisphosphate. It just has a dumb name. It is what it is. Okay. Most of you aren't even gonna take chemistry, so I'm not even gonna explain it. So it is what it is. But what do we do afterward? Go ahead and read it for me, please. Emily. Fructose 1,6-bisphosphate.
Okay. Fructose 1,6-bisphosphate is highly unstable and is cleaved to produce 1 hydro.
Just read the names in parentheses. Instead of
1 DHAP
and
1 G3P.
so fructose 1,6-bisphosphate, what we just made by adding a phosphate at the front, phosphate at the back, is highly what? So we have to, what does cleave mean? Like the cleavage furrow. We have to cut it. We have to split it. So we do this. We end up getting this because we what? Cut it, we split it, we cleaved it. But this guy, his name is, again, like, don't even worry about the whole name, we're just using abbreviations. DHAP, this guy is called G3P. For G3P, we're gonna turn it into pyruvate. But to actually get that, or to get pyruvate, which is good for us, we actually end up getting a lot of good stuff. We get an ATP here, we get another ATP here, and we get something called NADH. Again, anytime we break something, that energy is released and stored in this compound called NADH. So, one more time. We start off with glucose, we add a phosphate at the, and a phosphate at the, now it's called fructose 1,6-bisphosphate. It's highly what? And so we have to cut it, cleave it, split it. So we get this guy, and we get this guy. This one can go straight into making what?
Pyruvate.
And we end up getting
2
2 ATP and a what?
NADH.
But this guy though, he's a little bit different because we don't really want this one. So we have to use it and turn it into G3P. And we do that with an enzyme called,
Isomerase.
because remember isomerase is that deals with isomers. Isomers have the same blank, but different blank. Same formula, but different structure. Okay, so we have to convert it to G3P. But then G3P can be converted into what?
Pyruvate.
Pyruvate. Just like here. And again, what do we get out of it?
2 ATP.
2 ATP and an NADH. So the whole point is we add, one more time, we have glucose, we add a what? Phosphate at the front, phosphate at the, we create fructose 1,6-bisphosphate, which is highly,
Unstable.
and we
Cut it.
cleave it, split it to produce G3P and
DHAP.
G3P is the good one, we want it, and we convert it into,
Pyruvate.
and when we do that, we get
2-ATP.
N,
NADH.
perfect. But DHAP though, we gotta convert it into using,
Isomers.
and what do we get out of it? Once we get G3P, immediately we can make what?
Pyruvate.
Pyruvate, which we produce
2-ATP.
and a
NADH.
So the end goal of this was just to make what?
Pyruvate.
Pyruvate. So we got that. I think it's question number 8. This is what are the products? Right?
Mhm.
So what are the products of glycolysis?
ATP,
How many ATPs?
2, NADH,
How many ATPs?
4,
4 ATPs. And how many NADHs?
2,
How many pyruvates?
2.
2. Now, before we move on to the next part, I think it's question number 7. What does it say? Abigail, can you read for me, please?
What is the net gain of the ATP molecules from glycolysis per glucose molecule?
So this question is a little bit different because it's using the word net total. So what is the net gain or net total of ATP produced?
The only entire process.
So how many did we get?
2 in total.
So I think that's what Matthew was saying when he was saying 2, because we think about it, whenever you, like, if you get, like, work somewhere, I don't know if you work, but if you do, let's just say you get paid $10 an hour, you work 10 hours, how much should you get paid? But is that how much you actually get in your check? Because that's the total pay, that's not the net pay or gross pay, right? What's the net pay? How much you actually make after taxes? So here, what are our taxes? 2 ATP, we make 4, but it costs us 2. So what is the net ATP? 2. So again, these are questions that are on the exam. The question is, what is the net ATP produced in glycolysis? Answer? 2. What is the total ATP produced in glycolysis? Answer? 4. Total, the answer should be 4. Net, the answer should be 2. Question?
And I'm just happy you said that.
What?
That was gonna be on the exam.
All of these questions and the questions that are on the review, the questions that are on the right side, and the questions that are on the Kahoot are all direct questions from the test. Okay? I don't really do that often, but this is, like I said, there is no critical thinking involved. It's literally, can you memorize it? And it's just straight up facts. What is produced? Where is it produced at? That's basically, that's all the questions are. Yes.
What happens to the 2 ATPs that are paid in taxes?
So those ATP, well, actually, I'll show you in just a second. So in this right here, we start with glucose. It costs us what?
2 ATP.
And so we can see it costs us 2 ATP, but it produces what?
ATP.
ADP. So those ADPs are just sitting there.
Oh.
They just wait to gain another phosphate to make ATP again. Okay?
Okay.
So we get fructose 1,6-bisphosphate. It's highly what?
Unstable.
So we make
DHA.
and G3P, again, the full names, don't gotta worry about it, just the nicknames. Then we convert them both into
Pyruvate.
pyruvate. And how many carbons are pyruvate? 3. But we end up making 2 ATP and an ADH. For a total of how many ATPs?
4.
Or a net total of 2. I think we did 8 and 7, so go ahead and look at 5 and 6 real quick. So number 5 says, where does glycolysis occur? In the,
Cytoplasm.
if you look right here, if it's in the pink box, it's the cytosol. But cytosol and
Cytoplasm.
are the same thing. Okay. Number 6, during glycolysis, glucose is split into 2 molecules of
Pyruvate.
pyruvate. Good. Okay, let's move on. So go ahead and read it for me, please. This is called pyruvate oxidation, and that word is acetyl-CoA. So Itzel, go ahead and read it for us, please.
Pyruvate oxidation occurs within the mitochondrial matrix, and its purpose is to convert pyruvate into acetyl, CoA.
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 So one more, just a second. So we have pyruvate that is how many carbons? 1, 2, what? 3. And we're gonna convert it into acetyl-CoA, which is how many carbons? It's just 2 with coenzyme A attached to it. That's all it is. So this whole process is really short and simple because all we're doing is taking 3 and we're cutting a what? What are those little circles? Carbon. So we cut a carbon because it's only how many now? 2. That's all we're doing. And we're adding this guy over here too. Okay? So just FYI. And so go ahead.
Pyruvate, a 3-carbon compound is brought into the mitochondrial matrix where it is decarboxylated, meaning it
Good.
loses 1 carbon, which is released as carbon dioxide.
So what happens? Pyruvate is decarboxylated. But what does that mean? We just cut off a what? Carbon. This was 3, this was what?
2.
So we cut off a carbon, one right there, and that carbon is lost as what? Carbon dioxide. So where do we see carbon dioxide in our bodies? What we breathe out. So why do we breathe out carbon dioxide? Why does your pets, why does a fish, why does a turtle, why does the random raccoon in the middle of the forest, why do all of those things breathe out carbon dioxide? Because it's a waste product in this step. Okay? The carbon dioxide that we breathe out was once glucose that we consumed. Okay? So this is CO2. But since we broke something, all that energy is stored in the form of NADH. Again, we broke a carbon, the energy was released and stored in NADH. Okay? Again, we'll have the pictures in just a second, but this is just my other version of it before we get to the other picture. So you can kind of see what we're talking about step by step. Okay? Go ahead and read the next part for me, please. Isabel?
The remaining 2 carbon fragment is oxidized and NAD is reduced to NADH, capturing high energy electrons.
Right there. So we see this word oxidized and reduced, or oxidation and reduction. So oxidation is losing what?
Electrons.
While reduction or reduced is there's another, you can use oil rig as a way to remember it, or Leo says Berg. So losing electrons is oxidized, gaining electrons is reduced. It's just either way, it's perfectly fine. Also on the exam, I'll ask, for example, to go from, I'll just NAD plus to this. I'll have that, and I put, was this reaction oxidized or reduced? A is oxidized, B is reduced. So what happened? Did it gain something or lose something? It gained, right? So what is that? Reduction. So it would be reduction. Okay. Again, you'll see these again when we do the group next week. Okay. Go ahead and continue reading on, Isabella, please.
The 2 carbon molecule attaches to coenzyme A, CoA, forming acetyl-CoA. This occurs twice as there were two pyruvate molecules producing glycolysis. Acetyl-CoA is now ready to enter the citric acid cycle.
So, one more time, pyruvate is decarboxylated, which means that we cut off the what?
Carbon.
And it's lost as
CO2.
CO2. And then when we do that though, we produce a
NADH.
NADH. This CoA just glues to those two carbons that are left over to produce what?
Acetyl-CoA.
Acetyl-CoA. Again, this CoA is just glued to it, and it's like a taxi. It's gonna take these two carbons, and it's gonna deliver it to what?
The citric acid.
The citric acid site. It's just gonna take it to the next step. Again, it's a taxi, it's an enzyme, it's a taxi, it's just going to deliver it, drop it off, go back, pick up another two carbons. Take them to the citric acid cycle, drop them off, and then it's gonna do what? Then go back, pick up these other two carbons. Then it's gonna take them to the what?
Citric acid cycle.
Drop them off, and go back. It's a coenzyme. Enzymes, just like as we mentioned before, are never used up, they're just recycled. They're just a taxi. You drop them off, pick them up, drop them off, pick them up. That's all they do. Okay? So, let's go ahead and look at the steps. So, again, pyruvate is decarboxylated, which means we remove A in the form of
CO2.
CO2. And those two carbons, what do we do with them? We glue the CoA or coenzyme A to it to make acetyl-CoA. But since we broke something, again, we release that energy in the form of
NADH.
NADH. Okay. And it happens twice because how many pyruvates did we have?
2.
Because we made two pyruvates in what step? Those are the G,
Glycolysis.
glycolysis. So go ahead and answer questions 9 through 12, please. You could also ask your shoulder partners for help if you need. Also for assignments. Number 9 says pyruvate oxidation takes place in the mitochondrial matrix. So if you look right here on page 8.4, the first step, glycolysis, happens outside of the mitochondria. It happens in the cytosol or the cytoplasm. Then this is the outer membrane, this is the inner membrane of the mitochondria. So all of this is inside of the mitochondria. So that means pyruvate oxidation in the next step, citric acid cycle, actually happens inside of the mitochondria. Does that make sense? Okay. That's why I did this picture so you can see it as kind of like where it's going on. Then we have, number 10 says, what molecule is formed when pyruvate is oxidized and attaches to coenzyme A? Acetyl-CoA. Acetyl-CoA. And number 11, during pyruvate oxidation, each pyruvate molecule loses one carbon in the form of CO2. Again, this is where every CO2 that we breathe out, this is where it's coming from. This and the next step as well. Okay. Number 12, what are the products of pyruvate oxidation? Acetyl-CoA. How many? Two.
NADH.
How many NADHs? Two. And is that it?
CO2.
CO2, how many? Two. Good. Again, you notice how a lot of this stuff happens twice. Okay. So let's go ahead and look at the citric acid cycle, but before we do, I want us to kind of see kind of what's going on. Okay. Again, I know it's a lot, but baby steps. Okay. What happens with acetyl-CoA again? The CoA does what? It leaves to the, and goes back to the previous step to pick up another what? Another two carbons, and then it takes it to here, drops it off, goes back again and again and again and again, okay? But this is two carbons, and this is how many carbons? Four. This compound is what we call, actually, so 2 plus 4 equals what? Again, don't worry about names, just 2 plus 4 equals what? 6. So if we have 6 carbons, how many does this one have? 5. So what did we do? We lost one. Again, what does that mean? Starts with a D. Decarboxylated, because we removed a what? A carbon. But how is that carbon lost as? CO2. But again, when we break something, that energy is released and stored in a, called what? NADH. One more time. What happens to the CoA? It leaves, it goes back and picks up another 2 carbons. So this 2 carbons and this 4 carbons gets what? Combined to form this guy that has how many? 6. Then what do? We decarboxylate it, which means we lose a carbon, but that carbon is lost as CO2. So how many do we have now? 5. But when we break something, that energy is released and stored in the form of NADH. Okay? But how many do we have now?
5.
Do we keep it as 5?
No.
How many is this one?
4.
So what do we do to this carbon? We, same thing, decarboxylate, which means we remove it, and it's lost as? And since we broke something, the energy is released and stored as NADH. And how many do we have left? 4. But this 4 is not the same as this 4. Okay? So we do a whole bunch of chemical reactions that we don't even have to know. We just need to know that stuff happens, good stuff, to turn it back into this thing over here. Okay? So let's go ahead and go back and look at these different steps. So go ahead and read it for me, please. Matthew.
Which one?
Starting with the citric acid cycle.
The citric acid cycle, also referred to as Krebs cycle, occurs in the mitochondrial matrix, and its purpose is to break down acetyl-CoA, capturing energy in the form of ATP and electron carriers. Acetyl-CoA, 2-carbon compound, combines with
So this one's a little bit harder. It's right here. Oxaloacetate. Oxaloacetate.
4 carbons to form citrate or 6 carbons, and coenzyme A, CoA, separates and returns to the previous step. Remember that enzymes are never used up in the chemical reaction, they're just recycled.
Right there. So again, what happens? Acetyl-CoA, which is 2 carbons, gets glued to this guy over here called oxaloacetate, which is how many? 4, to make up a new compound with how many?
6 carbons.
This is called citric, like the citrus. Citrate like citrus. But this 6-carbon compound is ready to do whatever it's gonna do. Okay? Go ahead and read the next part for me, please. Soraya. Citrate is decarboxylated.
Citrate is decarboxylated and produces a 5-carbon compound along with the CO2 molecule, and NAD plus is reduced to NADH.
So NAD plus is reduced, but in other words, NAD is gaining electrons, that's why it becomes NADH. But citrate was how many carbons? 6. We decarboxylate it, which means we remove 1, and it's lost as CO2. But since we broke something, the energy is released and stored in NADH. Does that make sense? How many do we have now? 5. So right, go ahead and continue.
The 5-carbon compound is decarboxylated and produces a 4-carbon compound along with the CO2 molecule, and the NAD plus is reduced to NADH.
So same thing, 5 carbons, but we decarboxylate it, we remove 1, it's lost as CO2, and since we broke something, the energy is released and stored in the form of NADH. Okay. Go ahead and continue for me, Christian.
Carbon compound is not the same as the original oxaloacetate
Oxaloacetate.
and undergoes a series of chemical reactions to produce an ATP. NAD plus is reduced to NADH and FAD is reduced to FADH2.
So look at this one. So acetyl-CoA, the CoA leaves. 2 carbons plus 4 carbons make 6, which is who? Citrate, like citrus. Citrate is decarboxylated. Energy is broken or released and stored in the form of NADH. Carbon dioxide we breathe out. That 5-carbon compound is decarboxylated again. Carbon's broken off, releases CO2. Since we broke something, energy is released and stored as NADH. But we end up getting these 4 carbons. But as we said, this 4 carbons is not the same as what? Those 4. So we have to do stuff to turn it into oxaloacetate again. What stuff? Doesn't even matter. You just need to know that it goes in, that we make it. Okay? But when we make it, we end up getting what out of it? That's what we get out of it. So again, this 4-carbon compound is not the same as this 4-carbon compound, so we have to make it. But when we make it, we end up getting good things out of it. We get an ATP out of it, and we get 2 electron buses or electron carriers out of it. Okay? Does that make sense? Somewhat? I'm glad that Abigail's recording it and that she's gonna put it on YouTube.
You're gonna have to subscribe to me.
She says she's just gonna upload them because it's easier.
Yeah, I feel bad because no one knows how to get them. And I haven't even watched them because I feel guilty because no one has them, so I don't know.
Well, that's very kind of you.
I suppose.
But this happens twice because there are 2 acetyl-CoAs. Why are there 2 acetyl-CoAs?
Because there was 2 pyruvates before.
Because there were 2 pyruvates that we made. And why were there 2 pyruvates? Because we split the glucose. Okay? So go ahead and answer questions 13 through 16 real quick.
So, can I use the restroom?
Yeah, go ahead. Number 13 says, how many times does the Krebs cycle turn per glucose molecule? 2 times, twice. Number 14, in the Krebs cycle, acetyl-CoA combines with oxaloacetate to form citrate, like citrus. Citrate, like citrus. Which of the following is not a direct product of the Krebs cycle? Glucose. Why does it keep saying Krebs cycle?
Because it's the same thing as the citric
It's
acid.
the same thing as the citric acid cycle. Same thing. Again, it's just 2 names. Okay. Krebs was the name of the guy who discovered it. Okay. And it's called the citric acid cycle because we use citrate. Okay. Number 16. What are the products of the citric acid cycle? I mean, in total. How many NADHs do we make? 2. Do we? How many NADHs do we make?
3.
6 because this happens twice. There we go. How many CO2 do we make?
4.
Because it happens twice. And then how many FADHs 2s do we make? 2. Because it happens twice. But there's 4 blanks. So what is the last thing we make? ATP.
Oh, it's ATP.
How many ATPs? 2. So technically we really don't make oxaloacetate or the CoA because they're just recycled. So it's not really, because even though we make it, it's used back up as a reactant. So it doesn't really count. At least I'm not going to consider it. So you guys have to worry about that. Because I want it to be like the same products for each of these different reactions. Pyruvate, acetyl-CoA, the NADH is ATPs. So it's like the same thing over and over again. You just have to know like how many are made in each step. So before we look at this one, because this one is quite a bit, I want us to look at this picture real quick, okay? And I'm gonna give us this as an analogy. So 1, 2, 3, 4, 5. Okay, so Abigail, scoot down one.
Like,
Or Samuel will
sit
back.
and sit.
Yeah. And then I want you guys to just watch what they do, okay? And you guys turn this way. Okay, that's perfectly fine. You don't have to turn all the way around. Okay, so the whole point of this is there are 5 proteins on the inner membrane. 5 different proteins. And the whole point of this whole thing, pyruvate oxidation, acetyl-CoA or glycolysis, and citric acid cycle, was to produce electron carriers, electron buses. But we end up getting these 5 right here. So what we're gonna do is, I'm gonna give them to Nicholas. He's the first one. He's gonna pump across a hydrogen, put it right here, and then give the rest to Christian. So pump across a what? Hydrogen. Give the rest of the energy to Christian. Christian's like, oh cool, and then give it to Caitlin. Caitlin's gonna take some energy and pump it across as a hydrogen, and it's gonna give it to Emily. And then Emily, you're gonna be like, okay, cool, and then you're gonna give the energy to Abigail. She's the last protein in this step, which is this yellow one. But since she's the last protein, how much energy you got?
At 3.
You have 3. So what is she gonna do with it? 1, she's gonna pump across, and I'm oxygen. I'm like, hey, 2 hydrogens. But what's oxygen plus 2 hydrogens make? Water. Does that make sense?
Yes.
Even though what we're about to read doesn't sound like that, that's all that's happening. These proteins, one of them pumps across a hydrogen, that electron in the form of hydrogen, gives the rest of the energy to the next guy, then gives it to the next guy. They take the energy, pump across a hydrogen, give it to the next guy. Takes it, gives it to the next guy, and then pumps across a hydrogen. As we just saw, Abigail was the last one with how many energies?
3.
3. She pumps across 1, but she still got 2. Who did she give it to?
Oxygen.
Oxygen. This is why we breathe oxygen. We breathe oxygen, fish breathe oxygen, every living thing on earth breathes oxygen or requires oxygen because oxygen is the final electron acceptor. is going to end up making water. Okay? Okay. So let's look at it. So, what does that look like? Well, is this whole thing right here, okay? So, Caitlin, go ahead and read the first sentence for me, please.
The electron transport chain occurs on the inner membrane of the mitochondria along the plasma membrane of prokaryotes. And its purpose is to use the high energy electrons from NADH and FADH2 to create a proton gradient that drives the production of ATP.
Right there. So again, all it's doing is using those electron carriers, those electron buses. It's using those electrons to basically just pump across hydrogens. That's the first part. We're just pumping across hydrogens. Okay? Go ahead and continue reading for me, please. Nicolas? The process works by the third sentence.
The process works by NADH donating its electron to NADH.
Dehydrogenase.
As its being donated, the energy from the electron are used to pump across an H plus proton into the intermembrane space of a mitochondria.
Right there. So again, right here, this is what we're talking about. The energy is being given to this orange guy, which is NADH dehydrogenase. They take that energy and pump across as a what? Hydrogen. The energy that's left over, it gives it to this green guy, which is called ubiquinone. Okay, they all got weird fancy names. Okay. Go ahead and continue, Nicholas. The electron is then fifth line. The
The
electron
electron is then transferred to
ubiquinone. Ubiquinone.
ubiquinone.
Mm-hmm.
Also where FADH2 donates its electron, which are carried to the BC1 complex.
Perfect. And what is the BC1 complex? Go ahead and read it for me, please. Sam.
BC1 complex uses the energy from the electrons to pump across the H proton into the inner membrane space. The electrons are then transferred to cytochrome C, where they are carried to the cytochrome oxidase complex.
Right there. And Abigail, finish that last sentence.
Reduces.
Cytochrome oxidase.
Oh, wait. Cytochrome oxidase complex reduces oxygen. It gains electrons to form water and pump across H positive proton into the intermembrane space.
So again, the whole point is we're getting energy to pump across a what?
Hydrogen proton.
Hydrogen. Again, NADH. That H that we got is being pumped across into the intermembrane space. FADH2, those hydrogens are being pumped across into the intermembrane space. Okay? Just as we saw, like that last row, that's the intermembrane space. Okay? But what do we notice in the intermembrane space? There's a very high concentration of who? Hydrogen. While here, there's a very low concentration of hydrogens. Just like a dam, this protein, which is called ATP synthase, what do you think it does? It synthesizes ATP. But how does it do that? It does that by, again, like a dam, instead of water, it's forcing what to go through? Hydrogen ions. And as it does that, it's using that energy from the hydrogen ions, just like how a dam uses the energy from water moving to make what? ATP. It's like, hey, you can go ahead and go through, but as you go through, I'm going to use that energy to make ATP. That's the whole point. We use glucose, we cut it up, and whatever's left is just electron carriers, NADH, FADH. We use that energy, the electrons, to pump across what? Hydrogen ions. We use those hydrogen ions to create a gradient, high versus low. A lot of water, a little bit of water. And force it to go through this blue thing, which is called ATP synthase. So we can make it and turn it and force it to produce ATP. That's the whole point. Again, every single living thing on Earth does it exactly like this. Okay? So, we're gonna read the last part, and then I'm gonna leave it on that picture, and then we'll kind of wrap it up a little bit, okay? If you would go to start with the higher concentration, please, Savannah. The second paragraph.
The higher concentration of hydrogen protons in the inner membrane space leads to chemiosmosis, where protons naturally want to move back into the matrix, where there is a low concentration. ATP synthesis takes advantage of this and allows the hydrogen protons to flow through it.
Right there. So, chemiosmosis. It's like osmosis, but not with water, with hydrogen protons. Okay? Emily, go ahead and continue on, please.
Harnessing the energy to add a phosphate to adenosine diphosphate to produce ATP. This process can produce about 32 ATPs from one glucose molecule.
So we get quite a bit of ATP produced. Do you guys remember what is the total or the net ATP produced in glycolysis?
2.
2. How many ATP are produced in pyruvate oxidation? Look at pyruvate oxidation, which is question number 8. Question number 8 off to the right. How many ATPs were produced?
4.
ATPs? What is that? Wait, never mind. Number 12. How many ATPs are produced? None. So pyruvate oxidation produces none. How many ATPs are produced in a citric acid cycle, which is question number 16? 2. So we produce 2 in the first step, 2 in the third step, which is very, very little, but how many do we get in the last step? 32. That's the whole point. We save it all up for this big step so we can actually make ATP out of it, okay? So go ahead and we're gonna answer these questions and then I'll go back to this picture, okay? So, actually, I'm just, yeah, let's do 17 through 20, or 19, 20-ish, and then we'll go back to the picture. The final electron acceptor in the electron transport chain is the final electron acceptor in the electron transport chain is, remember these are transferring all of these energies. Who's the last one? O2. Again, this is why we breathe oxygen, because oxygen is the final electron acceptor. Because it's actually gonna make what? Water. Number 18 says, what is the primary role of the proton gradient created by the electron transport chain? So, sorry, I said to drive the production of ATP via ATP synthase. Does that sound good? Good. 19. Approximately how many ATP molecules are produced by the electron transport chain and chemiosmosis per glucose molecule? 32. Number 20, match the following terms with their descriptions. The process that produces ATP by utilizing flow of protons.
ATP synthase.
ATP synthase. Well, actually, no, because it says the process, not the enzyme.
ATC.
Proton gradient?
It's the whole thing.
ATC.
ETC, which is electron transport chain, yes. Wait, we'll come back to that. Let's do C. C says the enzyme that converts ADP to, so who actually does it? ATP synthase, good. D says the difference in proton concentration across the membrane. Proton gradient. And then B says a series of protons that pass electrons and pump protons. That one is electron transport chain, which is, yeah, so ETC should be B, because it's a series of proteins, excuse me. A says the process that produces ATP by utilizing the flow of protons. That one should be chemiosmosis. Okay, so again, I'm gonna go back to this. You guys go ahead and just finish filling this out. So we have about 15 minutes. I'm gonna let you guys finish this one, and after that, you can finish doing this one as well. Okay, all we're doing is just wrapping this up. Okay? I'm just giving you guys time to decompress and let it marinate.